Hello everyone, my name is Pallavi and today we are going to start with a very important chapter of grade 12th biology, which is molecular basis of inheritance. As we know that Mendel said that there are factors which are inherited from one generation to another and they are going to control your phenotypic characters, phenotypic trait. And later on we came to know that these factors are genes only, and we know that genes are this segment of DNA. So the specific segment of DNA, they express themselves and then they control a specific phenotypic trait. That was a gene. So what in this chapter we are going to study about what is the chemical, the molecular basis of this genetic material of the DNA or the RNA. That's why the chapter name is molecular basis of inheritance. So Phil, I can say that in all the living organism, the genetic material is a nucleic acid. That nucleic acid can be a DNA, deoxyribonucleic acid, or an RNA, ribonucleic acid. In major of the organisms, in majority, DNA is the genetic material because it is more stable, and we are going to study about that why it is stable also. RNA is genetic material only in few viruses, like in your coronavirus or your tobacco mosaic virus as well. So there are two types of nucleic acid. Generally, we say that nucleic acids are polynucleotides. Poly means many. So there are many monomers of nucleotides which are going to combine and they are going to form a polymer of nucleic acid. So why not to study a detailed structure of a nucleotide? So let's just discuss what is the structure of a nucleotide. So nucleotide have three components. It is made up of nitrogenous base, a phosphate group, and a pentose sugar. Nitrogenous bases are of two types. They can be purines or pyrimidines. Purines include adenine and guanine, and pyrimidines include cytosine, uracil, and thymine respectively. Purines are nine-membered. They form nine-membered ring. Pyrimidines form six-membered ring. Six-membered ring of nitrogen and carbon, and here nine-membered ring of nitrogen and carbon. So if I am going to draw a structure, a common structure for a purine, so it will be like this, that a six-membered ring, and with that a pentose. So here if you will count, so what I can see here, that 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 members are there. And out of these 9 members, here, 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 and here, nitrogen groups will be present. On the other hand, if I'll draw a pyrimidine structure, I will draw a simple hexagonal ring, and in this hexagonal ring, I will have my nitrogen groups here and here. So this is a common structure of a purine and a pyrimidine. Coming to the second component, that is phosphoric acid or your phosphate group, which is PO4 3 negative. If I'll expand the structure, it will be like this, and 4 oxygen. And the third component is pentose sugar. Pentose sugar are again of two types. It is either ribose sugar or a deoxyribose sugar. See, ribose and deoxyribo, they are five carbon ring, five membered carbon ring. So if I draw a ribose structure, it will be like oxygen group, alright, and like this. So this is my carbon first, carbon second, carbon third, carbon fourth, and there will be a group. So this is my fifth carbon. So let's just label it. This is 1, 2, 3 dash, 4 dash, and 5 dash. Again, the basic structure of deoxyribose is same. Here also, this is my 1 dash, second dash, third dash, 4 dash, and this is the fifth carbon. So this is 5 dash. So where is the difference realize? See, here is ribose sugar and there the name is deoxyribose sugar. Deoxy means one oxygen must be absent. So if I'll mark here on the second carbon, students, here an H and an OH group is there. But in deoxyribo, there will be no OH. There will be only a hydrogen present. So one oxygen is absent. Here OH group is there. Here and H group is there. And this is one of the major benefit to deoxyribose sugar, that the DNA is stable, because of the absence of one oxygen it is less reactive, so the reactivity is less, the stability is more. So when these three structure arrange themselves in a specific manner, they are going to form a nucleotide. Here one thing you have to remember is that when nitrogen base combines with pentose sugar, it bonds with a specific bond which is N glycosidic bond. Glycosidic bond means it is going to form a bond with the sugar, and N comes from the nitrogen because this nitrogen groups are going to form a bond with the pentose sugar. So let's just see how these three components are going to arrange themselves. They are going to bind themselves and then they are going to form a polymer as well to form a specific DNA or an RNA. So let's just draw the structure here. So I have arranged the three components of a nucleotide and I have formed a complete nucleotide. So this is my nitrogenous base, this is my pentose sugar, and this is my phosphate group. I have already told you that here and here, here and here are my nitrogen group. So see, if I'll start the counting of the members of this ring, 1, 2, 3, 4, 5, 6, 7, 8, and 9. So here I can see that this is the ninth member, which is the nitrogen, which is going to form a bond with the first carbon of my pentose sugar. And I said that when a nitrogenous base and a pentose sugar forms a bond, they form a N glycosidic bond. Here, if you will observe that on the second prime carbon, I have taken H. So this is a ribose sugar or a deoxyribose sugar. This is a deoxyribose sugar. Here on the fifth carbon, 5 dash carbon, my phosphate group is going to attach. And this bond is called as phosphoester bond. So if I include my sugar and nitrogenous base, they together form a nucleoside. And if I join a nucleoside with a phosphate group, this complete structure is my nucleotide. So now I have to join different nucleotide with each other so that a polynucleotide is formed. So let's just see how a nucleotide makes a bond with the other nucleotide. So now with the 3 dash end of this sugar, my pentose sugar, a new phosphate group is going to attach. So if I'll attach it here, region phosphorus, again with the with the phosphate group, a pentose sugar will form a bond. And here what is going to happen, a nitrogen base pair, a nitrogenous pair is going to form a bond. And again, this is going to be my N-glycosidic bond. This bond is my phosphoester bond. But what about this bond? This bond, which is between the phosphate group of the 3 dash carbon of a pentose sugar and the other 5 dash carbon of another pentose sugar. This is called as 3 dash 5 dash phosphodiester bond. So this, the monomer structure of a nucleotide, they are going to combine, they are going to bond with each other, and they are going to form a polymer, a polynucleotide. In this manner, this is going to be formed. So if I'll say this is my 3 dash carbon. So if I'll say this is my 5 dash carbon, so I'll say that this is the 5 dash end of this nucleotide chain. And here again with this 3 dash carbon, a new phosphate group is going to attach, and when it is not getting attached, it is free. So this is my 3 dash end of the nucleotide. So I have a polarity in a nucleotide chain which is 5 dash and 3 dash respectively. So they are going to form a chain, and then they are going to do different chains of a polynucleotide. They are going to combine, they are going to arrange themselves in a helical structure, which we are going to study further in the helical structure of DNA. So the structure of DNA was given by two scientists, James Watson and Francis Crick. So we generally refer this model as Watson and Crick model. They named the model as double helix model or structure of DNA. And they studied a very common DNA, B type DNA. They said that there are two strands of polynucleotides, and they are anti-parallel to each other, and there is a single axis, and these two strands are just coiling and they are forming a helix. So this is the axis, and the two strands are just coiling around in a helical manner. Here I have just represented these colorful structures as the nitrogenous bases, and these are the backbone. This I can say, these strands, the red and the black one, these are the backbone of DNA, which are formed by a pentose sugar plus my phosphate group. And these nitrogenous bases, they are perpendicular on the backbone and they are facing inwards. Are facing inwards. As I said that there are two ends in a polynucleotide, 5 dash end from the fifth carbon, 5 prime carbon, and the 3 dash end is formed by the 3 prime carbon. So let's say this is the 5 dash end of this polynucleotide, and this will be the 3 dash end. Similarly, here will be 3 dash and 5 dash end. If you'll observe, I have just marked the poles as opposite. Like this is the 5 dash end and this is the 3 dash end. So here I started with the 3 dash end because these two strands are they have specific polarity and they are anti-parallel to each other. So that's why I have done. The diameter between these two polynucleotide chain is 20 Armstrongs. The diameter is 20 Armstrongs. And they said that in each turn, a 360 turn, which is called as pitch, there are 10 pairs of new nitrogenous bases. So I'll say there are 10 base pairs present. And the length of this one pitch is 34 Armstrong. So I can write 3.4 nanometer. By dividing the number of base pair and the length of the pitch, I can calculate the distance between the two nitrogenous bases, like this distance between the two nitrogenous bases. So I'll simply divide them. So what was the length of the pitch? That was 34 Armstrong, and the number of base pair present was 10. So it comes out to be 3.4 Armstrong, or simply I can write 0.34 nanometer. Now I know the distance, the number of base pairs in a single pitch. So I can calculate the number of base pairs and the length of the DNA which is present in the single cell. So what I can write here is, so in our DNA, in a single cell, the total number of base pair are 6.6 into 10 raised to power 9 base pairs. And what is the length of the pitch which we are having was 3.4 nanometer. And if I'll convert this 3.4 nanometer in meter, so what I'll do, 3.4 into 10 raised to power minus 9 meter, simply. So I'll just multiply it with the total number of base pair in a single DNA. So 6.6 into 10 raised to power 9 base pair multiply by 3.4 into 10 raised to power 9 the length. So this cancels out and my length comes out to be 2.2 meter per cell. See, such a long DNA is packed inside a very small nucleus. Our nucleus have approximately dimensions of 10 raised to power minus 6 meters. So such a long DNA is going to coil itself and it's going to be packed inside the nucleus, and we are going to study the packaging of DNA as well. Apart from this, there were few features of the double helix structure which was given by Watson and Crick. So let's just discuss what were the silent features of this model. So here I have just partially drawn the diagram of the two strands of nucleotide. Here you can see this is my pentose sugar, this is the phosphate group, and here A and T and G and C respectively, they are just representing my nitrogenous base, purines and pyrimidines. Here I want you to remember that the number of purines and the number of pyrimidines always remain same, because purine forms a bond with the pyrimidine only. So how we'll know how which purine is going to form a bond with which pyrimidine? So I have just learned this with a mnemonic at girls college. So adenine always form a bond with thymine with two hydrogen bonds. So here I am going to draw two hydrogen bonds. And guanine will always form a bond with cytosine with three hydrogen bond. In the RNA, of course, the thymine will be replaced by uracil. So I can here again just draw like this. So adenine forms a bond with uracil in case of RNA. So here now I can draw the bonds. A and T will form two hydrogen bonds. Again, adenine and thymine forms two hydrogen bond. Guanine and cytosine form three hydrogen bonds. And as I said that the backbone of my polynucleotide chain, the backbone consists of a phosphate group and a pentose sugar, and the nitrogenous bases, they are projected inwards. So the first feature of this double helix model is that the DNA is made up of two polynucleotide chain. Yes, this is the first polynucleotide chain, this is the second polynucleotide chain, and the nitrogenous bases, they are going to project and facing inwards, and there will be main two strands of the backbone which are formed of one phosphate group and the pentose sugar. The second point is the two chains have anti-parallel polarity. Now I have already told you, see, this is this the phosphate group combines with the 5 dash carbon. So this will be my 5 dash end. And if I'll just move towards the end of this chain, this OH group is combined with at the 3 dash carbon. So this will be my 3 dash end. Similarly, here also this OH group is with the 3 dash carbon. If I'll count from this side, see here usually we have an oxygen group. So 1 prime, second prime, and third prime. So again, this will be my 3 dash end, and the phosphate group always bind with the 5 dash carbon. So this will be my 5 dash end. So if you will see, they are the polarities are anti-parallel to each other. So that's why I said the two chains have anti-parallel polarity, which means 3 dash to 5 dash end will be one chain, and the other chain will initiate with the opposite polarity, 5 dash to 3 dash end. The next feature was nitrogenous bases are paired with hydrogen bond. Abhi just we discussed that they are forming hydrogen bonds with them, and I have already told you that the number of purines will be always equal to the number of pyrimidines. Adenine will always form a bond with thymine or the uracil in the RNA, and guanine is going to form a bond with the cytosine. So if the number of purines and pyrimidines remain same, it helps in maintaining a uniformity, a uniform distance between the nitrogenous bases in a polynucleotide chain. The third point, sorry, the fourth point is, this point we have already discussed in detail very recently only. The two strands are coiled in a right-handed helical structure. The pitch of a helix is 3.4 nanometer and 10 base pair in each turn. The length is 3.4 nanometer and it consists of 10 base pair in each turn. And the distance between a base pair in a helix comes out to be 0.34 nanometer. We have just calculated this thing only. After that, one more point is the plane of one base pair stacks over other. This, in addition to the hydrogen bond, increases the stability of a helix structure. So this is one more reason why DNA is more stable. After these features, now let's just discuss a very important rule, Chargaff's rule, and on that basis few numericals are also there. So we are going to solve one example as well. You already know the features of a DNA helix model. So Chargaff's rule becomes very easy for you. Chargaff's rule was given by Erwin Chargaff, and he said that the purines and pyrimidines, they are always in equal number. So A plus G, my purines, and thymine and cytosine, they becomes equal. Also, he said that the adenine is going to always form a bond with thymine. Similarly, guanine will form a bond with cytosine, but adenine plus thymine is never equal to guanine plus cytosine. The third rule he gave is that the base ratio of the sum of adenine and thymine plus the guanine and cytosine, it never comes out to be same. It varies from species to species. So I'll write here, it varies from species to species, but it will always remain constant for a specific species, but it remain constant for a specific species. Now let's just solve one question on the basis of Chargaff's rule. The question is, if a double-stranded DNA has 20% of cytosine, so cytosine is 20% here. Calculate the percent of adenine. You have to find the percentage of adenine in the DNA. We have just discussed these 3 points and we are going to use these 3 points here only. So as I have already said, A plus G is equals to T plus में C. Can I say that if A plus G plus T plus and C, it will always be 100%? Yes. And then I already know that the value of C is 20%. And A is always going to be equal to T, G is always going to be equal to C, and if C is 20%, G is also 20%. So I am going to just replace the value here. I will write A plus 20% plus T plus again 20% equals to 100. And it comes out to be A plus T equals to 100 minus 40, it's 60. So what is going to be the value of A? A and T, they are equal. So I am going to divide 60 by 2. So A comes out to be 30%. And that's my answer. So very easy numerical comes from this topic. You just have to remember these three rules. Now there are few factual information regarding the DNA. Let's just discuss them. See, for the first time, Friedrich Mischer, he isolated DNA, but he was not aware that yeah, this is the DNA. He never named it as DNA. He just studied it, and it was not showing any character like a lipid or something else. So he just named it as nuclein. Later on, Altman identified the acidic nature of the DNA, so he just named it as the nucleic acid. One more thing, the length of the DNA is defined as the number of nucleotides or the pair of nucleotides, which is called as the base pair present in it. In E. coli, there are 4.6 into 10 raised to power 6 base pairs, and in an haploid content of human DNA, there are 3.3 into 10 raised to power 9 base pairs. Also, DNA is a heteropolymer. Now, why it is a heteropolymer? Because it is made up of different types, different components of a nucleotide. Let's just discuss the summary of this heteropolymer word. So as I said that the DNA is a heteropolymer, or I can say the nucleic acid is a heteropolymer because it is made up of different components of a nucleotide. What were the three different components of a nucleotide? One was a pentose sugar, other one was a phosphate group, and nitrogenous bases. So I have just mentioned different nitrogenous bases which are present in DNA or RNA, and what kind of nucleoside and nucleotide they are going to form. What was the nucleoside? Nucleoside was formed by the bonding of pentose sugar plus a nitrogenous base. So I have just mentioned different nitrogenous bases: adenine, guanine, thymine, cytosine, and uracil. And when they are going to form a nucleoside and a nucleotide, what will be their respective names? So if I'm going to mention a nucleoside, I generally add a suffix which is called as ine. So adenine will form a nucleoside which is called adenosine. For RNA and for DNA, I am just going to add deoxy in front of it. So it will be deoxyadenosine. For a nucleotide, what is a nucleotide now? When a nucleoside along with a phosphate group combines, it forms a nucleotide. For nucleotide, I generally add a suffix ate after the nitrogenous base. So it will be adenylate, or I can say adenylic acid as well. So for RNA, it will be adenylate or adenylic acid. For DNA, it will be deoxyadenylate or deoxyadenylic acid. Similarly, for guanine, guanine is guanosine. Again, I have added a suffix deoxyguanosine for DNA. guanylate or guanylic acid for RNA and deoxyguanylate. Now, see thymine. Thymine is not present in RNA. So, can a nucleoside thymidine exist in RNA? Never. So, thymidine never exists. In DNA, it is deoxythymidine. Again, in a nucleotide of RNA, thymidylate can never exist. A nucleotide of thymidylate can never exist because RNA don't have thymine. So only thymidylate will be present in DNA. Cytosine again, cytidine, deoxycytidine, cytidylate, deoxycytidylate in DNA and RNA. Now comes the uracil. Uracil is never present in DNA, it is only present in the RNA. So, can deoxyuridine be present as a nucleoside in DNA? Never. Only uridine will be present in the RNA. So, in a nucleotide, only uridylate, or we can say uridylic acid, can exist. Deoxyuridylate or deoxyuridylate acid can never exist. So that was for today's class. Whatever we have learned in today's class, I'm going to attach my handwritten note in the description. You can download and do the quick revision. And in the next class, we are going to discuss the packaging of DNA and different experiments performed to identify what is the genetic material in all the living organisms. Thank you for today. See you all in the next class.